validation. About this module. Follow the prompts on the screen to navigate through the module. Final assessment at the end of the module is your opportunity to demonstrate your knowledge, and you will receive a score on these questions. All answers have appeared in the learning material. Navigation tip. Screens are interactive. Navigation prompts may occur at the top or bottom of the screen. Objectives. The objectives of this e-learning presentation are to understand what is involved in the validation of processes, including process design, process qualification, and continued process verification, cleaning validation processes, analytical method validation. Validation qualification. Question: why do we validate processes and qualify equipment, systems? Answer: because the regulatory authority tells us we have to. No. Answer: because engineers are desperately in need of jobs. Wrong. What is validation? We validate processes and qualify equipment to reduce the variables in the manufacturing process. We qualify equipment to ensure that it operates as expected and within known tolerances. We calibrate instruments to ensure that the data being collected is accurate. We validate processes to define and verify the proven acceptance ranges, PAR, that produce a product that will meet specification. The PAR is defined by design of process understanding experience prior to verification by process performance verification. Validation versus qualification. Validation is establishing a documented evidence to provide a high degree of assurance that a specific system, process, or facility will consistently produce a product meeting its predetermined specifications and quality attributes. Processes, procedures, the way we use things, are validated, for example, cleaning, manufacturing processes, and analytical methods. Validation versus qualification. Continued. Qualification is a process of assurance that the specific system, premises, or equipment are able to achieve the predetermined acceptance criteria to confirm the attributes that it purports to do. Qualification is documented evidence that a specific piece of equipment, facility, or system is fit, ready for intended use. Things are qualified, for example, equipment, instruments, utility systems. Validation versus qualification. Continued. Example: the finished tablet fails dissolution testing. Root cause for the OOS: lab error, process error, equipment error, personnel error. Difficult to determine unless variables are reduced. Qualify, validate to reduce variables. Overview of validation qualification documents. Validation qualification support systems. Re-qualification program, schedule. Metrology program. Change control program. Preventive maintenance program. Process flow diagrams. Equipment lists matrix. References SOPs. Validation Master Plan, VMP. The VMP is a high-level document that establishes an overarching validation plan for the entire company or project to be used as guidance to company personnel or project team, and technical planning. Is a living document. Needs to be maintained. Evaluated annually at a minimum. Most important part is the schedule for validation and revalidation. Schedule should show all validation and qualification events. What date they occurred, what date for revalidation, when did revalidation occur. Validation Master Plan, critical elements. Validation lifecycle process. The activities in this process are executed by a cross-functional team, which includes subject matter experts, SMEs, from the impacted stakeholder departments. The requirement documents, user, functional, regulatory, etc., are generated by contributions from stakeholder groups. Risk assessment is conducted to identify critical aspects, CA, of the equipment and processes. Relevant documents are generated and executed per the verification plan, such as Factory Acceptance Test, FAT, Site Acceptance Test, SAT, Commissioning, IQ, OQ, and approval prior to starting the next activity. Content of the verification protocols depends upon the risk assessment as delineated in the verification plan. Validation Master Plan, critical elements continued. Personnel responsibilities, quality, engineering, production, validation, regulatory. Facility description, capabilities, scope of validation qualification activities, manufacturing, laboratory facilities, direct impact utilities, manufacturing laboratory equipment, computer software validation, cleaning validation, analytical, physical, and microbiological method validation, manufacturing process validation, description of protocols report, DQ, IQ, OQ, PQ. Validation Master Plan, critical elements continued. VMP should contain change control procedures, policy and procedure. Failure to properly document changes to the system means invalidation of the process. Software changes, controllers. Site changes, operational changes. Change of source material. Change in the process. Production area changes. Significant equipment change. Support system changes. Validation protocol. Written plan describing the process to be validated, including production equipment. How validation will be conducted. Objective test parameters. Product characteristics. Predetermined specifications. Factors affecting acceptable results. Protocol for validation of manufacturing process. Purpose and prerequisite for validation. Presentation of the whole process and sub-processes, including flow diagram and critical step analysis. Validation protocol approvals. Installation and operation qualification. Qualification reports including method, procedure, release criteria, calibration of test equipment, test data, summary of result. Protocol for validation of manufacturing process. Continued. Product qualification test data from pre-validation batches. Test data from formal validation batches. Sampling plan, where, when, and how the samples are to be taken. Evaluation of test data. Conclusion. Any need for requalification and revalidation. Certification and approval. Summary report of finding with conclusion. Copies of product stability. Life cycle approach. Process validation. Process design. Identification of process variables. Control strategy. Process qualification. Process monitoring and improvement. Validation report. The validation report contains executive summary, discussion, conclusions and recommendation, list of attachment. Topics should be presented in the order in which they appear in the protocol. Protocol deviations are fully explained and justified. The report is signed and dated by designated representatives of each unit involved in validation. Summary, validation. Validation and qualification serve a purpose. Reduce the variables in manufacturing. Use common sense. Don't overcomplicate things. Document why. Always provide justification for what you are doing. Do not qualify and validate in a vacuum. Use teamwork. Don't overlook the little guy. Process validation. What is process validation? The objective of process validation is to produce documented evidence that a specific process will consistently produce a product meeting its predetermined specifications and quality characteristics. The approach to process validation should be based on the overall product and process understanding. Continually assure that the process remains in a state of control during commercial manufacture. Collection and evaluation of information and data about the performance of the process will allow detection of process drift. Ongoing feedback about product performance is an essential feature of process control. The goals of process validation. Whether the process is effectively controlling the quality of finished product. Quality, safety, and efficacy is built into the product. Obtained consistent, reliable, and accurate data. Act as a proof in decision-making. Assurance of quality of product. Product liability, avoiding variability, controlling of monitor output, and validating process performance. Understand process, equipment, and system. Increase efficiency, shortening lean time. Lower the rejection and rework. Fulfill the regulatory and customer requirements. Types and stages of process validation. Types of process validation. 1. Prospective validation. It is establishment of documented evidence of what a system does or what it purports to do based on a plan. This validation is conducted prior to the distribution of new product. 2. Retrospective validation. It is the establishment of documented evidence of what a system does or what it purports to do based upon the review and analysis of the existing information. This is conducted in a product already distributed based on accumulated data of production, testing, and control. Concurrent validation. It is establishment of documented evidence of what a system does or what it purports to do, information generated during implementation of the system. 4. Revalidation. Whenever there are changes in packaging, formulation, equipment, or processes which could have impact on product effectiveness or product characteristics, there should be revalidation of the validated process. Conditions that require revalidation studies are changes in critical component, change in facility or plant, increase or decrease in batch size, sequential batches that fail to conform product and process specifications, stage, process design. The commercial process is defined during this stage based on knowledge gained through process development activities. 2. Process qualification. During this stage, the process design is confirmed as being capable of reproducible commercial manufacturing, including qualification of the facility, utilities, and equipment. 3. Continued process verification. Maintenance, continuous verification, and process improvement. Ongoing assurance that routine production process remains in a state of control, assessed by collecting and monitoring information during commercialization. Types of process validation. Prospective validation is the establishment of documented evidence of what a system does or what it purports to do based upon a plan. This validation is conducted prior to the distribution of new product. Types of process validation. Continued. Concurrent validation. Provide documented evidence to show that the manufacturing process is in state of control based on in-process monitoring of critical process steps and in-product testing of current production. Revalidation. Whenever there are changes in packaging, formulation, equipment, or processes which could have impact on product effectiveness or product characteristics, there should be revalidation of the validated process. Conditions that require revalidation studies are changes in critical component, change in facility or plant, increase or decrease in batch size, sequential batches that fail to conform product and process specifications. Stages of process validation. Process design. The commercial process is defined during this stage based on knowledge gained through process development activities. Process qualification. During this stage, the process design is confirmed as being capable of reproducible commercial manufacturing, including qualification of the facilities, utilities, and equipment. Continued process verification. Maintenance, continuous verification, and process improvement. Ongoing assurance that a routine production process remains in a state of control, assessed by collecting and monitoring information during commercialization. Summary of process validation. Process design. Verify equipment and utilities, build and installation. Verify operation in all operating ranges. Challenge in routine production, loads, interventions, and stoppages. Define critical attributes. Process qualification, PQ, combines qualified equipment and trained personnel with commercial manufacturing process, control procedures, and components to produce commercial batches. Continued process verification, continually assure that the process remains in a state of control. Continued monitoring at the level established during PQ until significant data is available to generate significant variability estimates. Success of process validation depends on information and knowledge from product and process development, controlling the manufacturing process to get desired quality attributes, understanding the sources of variation, detecting the presence and degree of variation, understanding the impact of variation on the process and ultimately on product attributes, controlling the variation in a manner commensurate with the risk it represents to the process and product. Process validation documents. Validation master plan. All validation activities must be described in a validation master plan, which must provide a framework for thorough and consistent validation of product. Key elements of validation are requirements, design control, testing, change control, traceability, and monitoring. Process validation master plans must include introduction with the scope of the plan, e.g., sites, systems, processes, responsibilities by function. B. Related documents, e.g., risk management plans, products, processes to be validated and or qualified. Validation protocol and report. The validation protocol must be numbered, signed, and dated, and must contain as a minimum the following information: objectives, scope of coverage of the validation study, validation team, their qualifications and responsibilities, justification for validation, risk assessment, type of validation, prospective, concurrent, retrospective, revalidation, a list of all equipment to be used with calibration, qualification, requalification, and preventive maintenance details, outcome of IQ, OQ for critical parameter, critical parameters and their respective tolerance, description of the processing steps, challenges of sampling and sampling plans, statistical tools to be used in the analysis of data, forms and charts to be used for documenting results, non-conference, out of specification, out of trend, deviation, change control, conclusion, summary, approval of study. Process validation lifecycle. Stage 1, prerequisite. Identify quality characteristics. Identify process parameters, in-process control, operating range and acceptable range, potential critical process parameter, lab scale experience, determination of ranges, references, specification, target product profile, determine upper and lower limit for operating and acceptable range. Stage 2, product process validation, manufacturing performance qualification protocol, in-process release and shelf life specification, additional consistency testing, approved validation protocol and report with test data and summary. Stage 3, life cycle management, create life cycle validation table, CPP, CQA, and in-process parameter control, maintain process technology, trend analysis and track the performance, periodic review of campaign batches. Process validation documents, documentation that verifies the validation activities such as validation master plan, validation protocol, report and summary, procedure test data, review change control, continued process verification. Cleaning validation, protocols approved by QC or QA and to cover, e.g., disassembly of system, pre-cleaning, cleaning agent, concentration, solution volume, water quality, time and temperature, flow rate, pressure and rinsing, complexity and design of the equipment, training of operators, size of the system. Protocols continued. The cleaning validation protocol should include objectives, responsible people, description of the equipment including the make, model, serial number, or other unique code, time intervals, bioburden, cleaning procedures, equipment used for routine monitoring, e.g., conductivity meters, pH meters, and total organic carbon analyzers, number of cleaning cycles, sampling procedures, e.g., direct sampling, rinse sampling, in-process monitoring, and sampling locations, and the rationale for their use. Protocols continued. The cleaning validation protocol should include 2. Data on recovery studies. Efficiency of the recovery of the sampling technique should be established. Analytical methods. Acceptance criteria with rationale for setting the specific limits, including a margin for error and for sampling efficiency. Cleaning agent to be used. Revalidation requirements. Protocols continued. The cleaning agent used should be scientifically justified and based on the solubility of the materials to be removed, the design and construction of the equipment and surface materials to be cleaned, the safety of the cleaning agent, the ease of removal and detection, the product attributes, the minimum temperature and volume of cleaning agent and rinse solution. Protocols continued. Bracketing. Very similar cleaning procedures for products and processes. No need for individual validation. Worst case may be acceptable and should be justified. Consider type of products and equipment, allowed only where products are similar in nature or property and processed on the same equipment, and identical cleaning procedures used. Protocols continued. Bracketing. Representative product, most difficult to clean. Equipment, only when it is similar or the same equipment in different sizes, e.g., 300-liter, 500-liter, and 1,000-liter tanks. An alternative approach is to validate the smallest and the largest sizes separately. Reports, the relevant cleaning records, signed by the operator, checked by production, and reviewed by quality assurance, and source data, original results, should be kept. The results of the cleaning validation should be presented in cleaning validation reports stating the outcome and conclusion. Equipment. Cleaning of contact surfaces to be validated with consideration to non-contact parts. Critical areas should be identified. Dedicated equipment for products which are difficult to clean, equipment which is difficult to clean, products with a high safety risk where it is not possible to achieve the required cleaning acceptance limits using a validated cleaning procedure. Equipment continued. For one SOP for cleaning a piece of equipment, review products being produced, cleaning in a large campaign, cleaning between batches of different products. The design of equipment may influence the effectiveness of the cleaning process. Consider design, e.g., V-blenders, transfer pumps, or filling lines. Detergents, released by quality control and where possible meet food standards or regulations. Composition should be known. Easily removed with rinsing, demonstrated with acceptable limits defined. Consider also possibility of detergent breakdown. Bioburden, prevent microbial growth and remove contamination. Equipment stored in a dry condition after cleaning, no stagnant water. Control of bioburden important. Sampling, general, clean as soon as possible after use, especially topical products, suspensions, and bulk drug, where the drying of residues will directly affect the efficiency of a cleaning procedure. Two methods of sampling, a combination of the two is the most desirable. Direct surface sampling. Rinse samples. Sampling, general, continued. Resampling, not to be done before or during cleaning. Constant retesting and resampling can show that the cleaning process is not validated, may indicate presence of unacceptable residue and contaminants resulting from an ineffective cleaning process. Direct surface sampling, direct method. Most commonly used method. Use swabs, inert material. Type of sampling material should not interfere with the test. Factors to be considered include supplier of the swab, area swabbed, number of swabs used, whether they are wet or dry swabs, swab handling and swabbing technique. Direct surface sampling, direct method, continued. Other factors include location from which the sample is taken, including worst-case locations identified in the protocol, composition of the equipment, e.g., glass or steel, critical areas, hardest to clean, e.g., in semi-automatic, fully automatic, clean-in-place systems. Use appropriate sampling medium and solvent. Rinse samples, indirect method. Allow sampling of a large surface, areas that are inaccessible or that cannot be routinely disassembled, provides an overall picture, useful for checking for residues of cleaning agents in combination with other sampling methods such as surface sampling. The manufacturer has to provide evidence that samples are accurately recovered. What is considered acceptable in terms of recovery? Recovery validation. A recovery of greater than 80% is considered good. Greater than 50% is reasonable. Less than 50% is questionable. Establishing acceptable limits. Limits should be practical, achievable, and verifiable. The rationale for them should be logical and based on knowledge of the materials. Each situation assessed individually. Principal reactant and other chemical variations. Establishing acceptable limits continued. The limit setting approach can be product specific. Group products into families and choose a worst case product. Groups according to risk, e.g., very soluble products, products with similar potency, highly toxic, or difficult to detect products. Use different safety factors for different dosage forms based on physiological response. This method is essential for potent materials. Establishing acceptable limits. Continued. Limits may be expressed as a concentration in a subsequent product, ppm, limit per surface area, mcg per square centimeter, in rinse water as ppm. Limits for carryover of product residues should meet defined criteria. Next slide. Establishing acceptable limits. Continued. The three most commonly used criteria are visually clean, no residue visible on equipment after cleaning, spiking studies to determine the concentration at which most active ingredients are visible, may not be suitable for high potency, low dosage drugs. No more than 10 ppm of one product will appear in another product, basis for heavy metals in starting materials. No more than 0.1% of the normal therapeutic dose of one product will appear in the maximum daily dose of a subsequent product. Establishing acceptable limits. Continued. The most stringent of three options should be used. Certain allergenic ingredients and highly potent material should be undetectable by the best available analytical methods, e.g., penicillins and cephalosporins, e.g., anovolent steroids, potent steroids, and cytotoxics. Dedicated manufacturing facilities needed. Analytical method validation. Introduction. Method validation is the process by which it is established through laboratory studies that the performance characteristics of the method meet the requirements for its intended purpose. It is a part of the overall validation process that also includes software validation, instrument qualification, and system suitability. Analytical methods should be validated unless the method employed is included in the relevant pharmacopoeia or other recognized standard reference. The suitability of all testing methods used should nonetheless be verified under actual conditions of use and documented. Analytical performance characteristics. Typical analytical performance characteristics used in method validation are as follows: specificity, linearity and range, accuracy, precision, repeatability, intermediate precision, reproducibility, detection limit, quantitation limit, robustness. Specificity. Specificity is the method's ability to measure unequivocally the analyte in the presence of the components such as excipients and impurities in the analytical matrix. For identification tests, specificity ensures the identity of the analyte of interest. For impurity tests, specificity ensures that the method allows for an accurate statement of the impurity content. For assays, specificity ensures an exact result for determination of the content or potency of the analyte. Methodology. If impurities and excipients are available, specificity is demonstrated by spiking the drug substance or product with the appropriate levels of impurities and determining them with the appropriate accuracy and precision. If impurities and excipients are unavailable, specificity is demonstrated by the ability to discriminate between compounds of closely related structures or by comparison to a known reference materials or perform an artificial degradation under drastic conditions, light, heat, acid, bases, etc. Linearity and range. Linearity. The ability of a method to produce results that are directly proportional to the concentration of analyte in the sample over a given interval, range. Range, the interval between the upper and lower levels of analyte that have been demonstrated to be determined with suitable level of precision, accuracy, and linearity using the method. Methodology, linearity, determined using a minimum of 5 concentrations, 3 samples per concentration is recommended. If there is a linear relationship, results should be evaluated by appropriate statistical methods. Range, determined by verifying that the method provides acceptable precision, accuracy, and linearity when applied to the extremes as well as within the range. Accuracy, the closeness of the results obtained by the method to the true value or the difference between results obtained by the method and the actual true value. Methodology, drug substance, evaluated by comparing the method results with the analysis of a reference material. Drug product, evaluated by analyzing samples spiked with known quantities of analytes or comparison of method results with that of a second well-characterized method. Impurities, evaluated by analyzing samples spiked with known amounts of impurities. Use a minimum of 3 replicates at 3 levels across the recommended ranges of 80% to 120% for principal components, 50% to 150% for impurities. Precision, the degree of agreement between results when a procedure is repeatedly applied to a homogeneous sample. Repeatability, intra-assay precision, precision under the same operating conditions for a short period of time. Intermediate precision, indicates intra-laboratory variations, different days, different analysts, different equipment, etc. Reproducibility, indicates interlaboratory variations, collaborative studies. Methodology, repeatability is determined by analyzing aliquots of a homogeneous sample and determining relative standard deviation. Recommended to use 9 measurements with the given range of the method, e.g., 3 concentrations per 3 replicates, or use a minimum of 6 replicates at 100%. Limit of detection, limit of quantitation. Limit of detection, LOD. The lowest amount of analyte that can be detected under the established experimental conditions. Limit of quantitation, LOQ. The lowest amount of analyte that can be quantitatively determined with acceptable precision and accuracy. Limit of detection, limit of quantitation, continued. Methodology, estimated using a signal-to-noise SN ratio. Limit of detection, SN greater than 3. Limit of quantitation, SN greater than 10. Verification, analyze suitable number of samples at or near LOD or LOQ, or estimated based on the standard deviation of the response and the slope. LOD equal 3.3 standard deviations per slope of calibration curve. LOQ equals 10 standard deviations per slope of calibration curve. S equals slope of calibration curve. Sigma equals standard deviation of the response based on standard deviation of the blank or the standard deviation of the y-intercept of regression lines or the residual standard deviation of the regression line. Robustness, a measure of the procedure's ability to remain unaffected by small but deliberate variations in the method parameters. Provides an indication of reliability of the method during normal use. Robustness is evaluated during development phase. If measurements are susceptible to variations in analytical conditions, the analytical conditions should be suitably controlled. A precautionary statement should be included in the procedure. Use the information to determine critical system suitability parameters. Use the information to establish acceptance criteria for validation study. Methodology, Purposely vary method parameters over a known range and determine the effect, if any, on the method results. Multivariate statistical experimental design can be used to control method variables. Analytical procedures and characteristics evaluated in validation. ICH Q2 R1. Final assessment. Review the module before commencing your final assessment. All questions relate directly to information and principles covered in the slides. You can only attempt each question once. Question types may include true-false, drag and drop, and multiple choice. Good luck.